So as the slides are coming up, I really don't, a lot of what I was planning to tell you, you've heard already. So we're just going to go through this quickly. The goal of this, and the reason why I was asked to speak at the end of this session, was because we anticipated some of the questions that you guys would have. And so we anticipated, how soon is this going to be ready? How do I get this therapy to my child? I think I can use this, right? How does this work? I got it. Never mind. Okay. All right, well, you work on that. So basically the goal of this was just to say, what is this process? Why does it take so long? Why can't I now give my child the oral medication or the gene therapy that I heard about today? So, okay. So this is just a little mouse who says that he can go home because the miracle drug cured him, but the people have to wait, which is frustrating. Now they're going on their own. All right, so we can skip the picture. This is basically what we're going to talk about. So we're going to talk about the process from bench to bedside, what it means to take a new idea, a discovery, a development, get it through preclinical studies, and then get to clinical trials. So we're going to start at the very beginning. So what happens when you have a big idea? These ideas come. So where did these ideas come from? So you heard a lot about some of the natural history work that's going on. You heard yesterday about some of the early seed grants, and know that NTSAD often funds very early research. And so the reason for that is that we know that when we learn more about these diseases, when we learn about the pathogenesis or what happens, what problems the disease causes, then that helps us really learn new strategies for how to stop the progression of the disease or how to prevent the complications of the disease. We also, as we learn about new technologies, learn new strategies, either of new therapies overall or new ways to deliver therapies. And then we have there's a number of mechanisms for studying either known molecules and approved molecules, or looking for newer molecules that may have effect in the disease. So once you have a new idea, then you have to find out, does it work, and is it going to be safe? So does it work? Does it work? Will it be safe? So does it work? We usually start with cell models and then with animal models to find out if giving the treatment idea is going to have any effect. And then also at the same time, they look for toxicity. So do the cells start to look ugly and do things that they're not supposed to do? When we treat animals, do the animals get sicker? Do the animals tolerate the therapy well? And then we can start to look for some effect in the animals. Do they start to get better? So once we get through animal models, then what happens? And you heard a little bit about this from Dr. Tift when she was speaking. So to get to a clinical trial is an extremely complicated process. So you heard about small animals like mice. You heard about bigger animals like cats. And sometimes you heard about finding even bigger animals that are closer to human size. So the idea is then that the FDA has to even approve beginning a clinical trial. So the FDA has to say, your data look good in the animals and in the cells, this looks to be safe, and then this looks like your plan is very reasonable. And so then the FDA is involved in that process of approving a clinical trial, and then the clinical trial goes through several phases that you've heard of already a little bit about. So this picture, which I hope for those of you who are here is a little bit clear, and this shows sort of as you go through this process, so in the beginning, what the timeline is. So in the beginning here is sort of getting started and all of the idea and the preclinical work in the lab, and then you go into clinical trials. And this is what happens after there's a clinical trial, which I think is something we can worry about for another day, because our goal for right now is to get to here. So as you heard a little bit, all early trials in most diseases are based really on safety. And a phase 1 study is really focused on safety and is usually in very small numbers. But for our diseases, we expect that phase 1 studies will be combined with some efficacy. You can go. And so how do we determine if a therapy works? And You've heard a lot of this already, so I don't have that much left to say. But we do know that it's very important that we be able to determine in small groups and in these patient populations whether or not the therapy that we're using is going to have any effect. So some of the work that you heard about earlier today and also yesterday is extremely important. You've heard this from Dr. Tiff, but I can just sort of, to give it another plug, that it's really important that we're able to understand what these diseases look like in the natural history and what the patient populations look like so we can know if we're having an effect. And then biomarker studies and imaging studies are also very important for that. So why is some of this so hard in these disease populations and in GM1? As you've heard, these GM1 isn't just really one disease, but there's a number of different subtypes and that those are variable. You've learned that even from what we used to think were the subtypes, we now learn that the subtypes can be further divided into sub-subtypes, and that even among patients who fit within a subtype, they have variability. So as Dr. Tiff said, sometimes for some studies, these patients may be their own control patients so that we can only compare them to themselves. We don't yet fully understand the natural history and the clinical features, but there's been a lot of good work to advance understanding this for GM1, and that we still have some work that's ongoing to determine what those endpoints would be. And then there's also some additional concerns when we deal with ethical concerns for rare diseases. So we know that families especially tend to have a higher tolerance for risk when it comes to therapies because we're dealing with life-threatening diseases that don't have any other available therapies, and that's, of course, very unusual. As you've heard, usually when a clinical trial begins, there's control patients, so patients who are not treated with therapies. We can compare them to those who are treated with therapy. There's obviously a lot of concerns about when we talk about diseases that are quickly progressive and life-threatening. There's also concern when we talk about some invasive procedures. So if we're talking about a therapy trial where you're doing intracranial injections into the brain, there's also ethical concerns on the other side. Certainly, I don't know that anyone would agree to have their child's brain injected if it wasn't certain that they were going to be getting a drug. So we're not doing, there's big concerns about intracranial injections of placebos. No one's really going to agree to that and shouldn't agree to that. There's also the idea of urgency, so we really don't want to delay treating people with life-threatening illnesses when we have the potential of a therapy. And so a lot of this is about really weighing the risks and the benefits, and that ratio may be a little bit different for families like yours who have less to risk and far more to gain. And I think this is the last one. So that you heard a little bit about this from Dr. Tiff, so she didn't know what I was going to tell you about. But this slide, these pictures here really compare phase 1 through phase 3 for a standard drug, I would use the same example she did, for example, a high blood pressure drug versus a therapy for GM1 or other rare diseases. So again, phase 1 trials for typical drug development usually involve somewhere between 20 to 100 healthy volunteers, people who don't even have the disorder, who take the drug just for safety purposes. And then they move into a larger study with several hundred patients to see if there's any effect, and if there's effect, then move into a third, and additional studies that may involve several hundreds to thousands of people to look for efficacy. That's obviously not something that's going to happen here. And I always like to point out that in the print that I know you can't read, that they're talking about orphan drug development. So for orphan diseases, which by definition in this country is diseases that affect less than 200,000 people. So compared to GM1, that's still huge, right? So GM1 is what we would call ultra-rare disease, much smaller even than an orphan disease. And so then we would expect that there would probably be a combined phase 1, phase 2 that would look for efficacy in addition to safety. But we think that the initial numbers are going to be very small. They're often fewer than 10 patients. And I think that's all that I have for you. That's all I got. Any questions? Or maybe no time for questions. 4:30 on the nose.
You're amazing.